This is CheloniaCast, episode 73. Today we are joined by Dr. Nicole Valenzuela, who is a full professor in the Department of Ecology, Evolution, and Organismal Biology at Iowa State University. She's also the principal investigator in the Laboratory of Evolutionary and Ecological Genomics at Iowa State University. She was also recently elected as a fellow of the American Association for the Advancement of Science, which is a high honor in any area of science, and has focused a lot of her career on better understanding sex-determining mechanisms in turtles and across vertebrates sort of largely, but her work spans an incredible amount of different areas of science focused sort of on the evolutionary ecological side of things. She co-edited the book Temperature-Dependent Sex Determination in Vertebrates, sort of this updated compendium on all different aspects of sex-determining systems, and has published over 100 peer-reviewed papers, many in top academic journals. Her work is really very interdisciplinary and focused on sort of ecology and evolution of a lot of different things, incorporating genomics, phylogenetics, and evolutionary and developmental biology to get at really complex issues, including sex determination, and many other areas. And so we're really excited to speak with her today about her history working with turtles, as well as sort of broader concepts in ecology. So thank you for joining us, Dr. Valenzuela.
Thank you for the invitation and having me here.
All right, so we like to start this the same way for everybody. So we just like to lead with, what like first got you interested in turtles, and then I guess more specifically for you, did your interest in like sex determining mechanisms come before your interest in turtles, or is that you kind of found along the way?
I think it happened both at the same time. I was doing research on other topics, and then I learned that turtles had temperature-dependent sex determination. In particular, I was working at this foundation, and they had done a little work with Podognomus expansa, the giant Amazonian turtle, doing some field experiments, sort of rudimentary, to look at their temperature-dependent sex determination. And they asked me whether I wanted to start a project looking at that issue more formally. I never worked with turtles up until that point, and I had no idea that temperature-dependent sex determination even existed. I was convinced from my undergrad training that all species have sex chromosomes. And so when I heard that, it was like I couldn't understand. Like, do you mean that some species, depending on whether it's warm or cold, they develop into a male or a female? That just bizarre. How haven't they gone extinct already? And is that why they are endangered? Because that giant Amazonian turtle happened to be an endangered species. So I went to the field. I designed a project to look at different constant temperatures and how they develop, how these embryos develop into males or females. And so I had to go to the field, collect the eggs for the incubation experiments. And when I just saw them out there in this riverbank nesting, I just fell in love with them. It was all of a sudden everything aligned, and it was like, this is the question I want to pursue for research, and this is the organism I'm fascinated with, and I haven't stopped.
That's awesome. Maybe we could just sort of start out with sort of diving into your work with the giant Amazonian river turtles, the Podocnemus. That's a really fascinating species, and you did some work looking at the early on kind of sex determination in P. expansa. And I'm curious, just one of the things that sort of stands out to me that could be difficult is hatchling turtles are challenging to sex visually. And so that's sort of, I mean, you can use the histology to sort of get at that question, but from a conservation perspective, that doesn't seem like it's very reasonable. So how did you get over that hurdle of figuring out what the sex of these hatchlings were after you had incubated them?
So that was the very first study that we did. So I did the incubation experiments with those eggs that I collected in the field. And then The founders of this foundation I was working with were friends with Valentin Lance from San Diego Zoo, who had done sexing of crocodilians and other species using testosterone levels in the blood. So he came to Colombia, where this study was conducted. He came to Colombia and taught me how to do sexing, looking at levels of testosterone in the blood. So we were also concerned that how are we going to sex them when we don't want to kill them. We just want to be able to find a way to sex them and then release them. So he knew how to do this. He came with sample blood from individuals and used what is called a radioimmunoassay, using some special way to measure the testosterone by using an antibody that has a radioactive marker. And so we had to find a laboratory that had a gamma counter to be able to measure radioactivity levels in the samples of the different individuals. So we presented that in at a meeting, and there's a little abstract that is one of my first publications in '92, so a while ago. And I used that same technique to sex turtles with temperature-dependent sex determination later on, and Podognomus expansa in particular later on. It's still a method that is a little bit cumbersome. Nowadays, there are alternatives to not having to use radioactivity, because you don't want to use radioactivity if you don't need to. And so there are ELISA measurements that is a similar idea, but instead of measuring radioactivity, you measure, it's a colorimetric. So there's a color change with chemicals that you put that bind to the testosterone, and then you can measure it. That still, we were trying to figure out, is there something else that that we can do with like shoestring budget, right? Very, very small amounts of money that people could do that would be easier for field biologists, so that for conservation in areas where these tests are still expensive and require a lab and require special equipment, anything else that was even simpler than that. And that's where we came up with looking at whether there were morphological differences between males and females. They, if you look at them to the naked eye, you cannot tell apart a male or a female from any baby turtle, right? But when you take a photograph and then you use what are called geometric morphometric methods that are much more sensitive to tell differences in the shape of the carapace we use first, by looking at the relative position of the intersection of the scutes in the carapace of a turtle. Then we were able to reveal that there were differences between males and females. They're very subtle, so you need these methods to be able to tell the animals apart. And you have to train the program first with a set of individuals of known sex that you have sex by either the testosterone level, or by laparoscopy, or a gonadal inspection of some sort. And then you could use that. It's still problematic if you, ideally what you want, and particularly for conservation, is to be able to grab an animal and say it's male or female in the cheapest possible way, non-lethal, the least invasive possible. And up to today, there is not a single protocol that allows you to do that, except for species with sex chromosomes. Then you can, because you can look at molecular markers. But for turtles with temperature-dependent sex determination, there's still not the silver bullet that allow us to do that. So the least invasive methods will be getting the blood to measure something like testosterone, or doing the geometric morphometrics. But still, I think better methods are still needed, and people are working on those.
Yeah, I guess it's sort of an advantageous when the Podocnemus hatch out. They're fairly large
Yes.
turtles just immediately out of the egg, and a
Yeah.
lot of species you can't. I mean, with a kinosternid,
Hmm.
good luck with doing that. They're so small, you'd have to wait for a long time. But
But we did this with a Chrysamis picta with painted turtles,
oh
and
wow,
they were small. They were, I think, 1 or 2 months, and they still remain very little. And we were able to use the geometric morphometrics. It has an error rate, like I don't remember exactly the figures, but maybe our accuracy was 85% or maybe a little bit higher for them, which is not bad, but it's better than nothing. But it's still not ideal, right?
that's interesting. I wouldn't expect that for something that small you could, but
I didn't either.
yeah,
When we did the study, I thought that we were going to be able to do it with the Porognimis, but definitely not with the Chrysamis. But we did. It did work.
that's interesting. Something else that's with the Podocnemus, they're
Mhm.
so interesting ecologically in terms of their nesting patterns, the sort of Arybata and clustered type nesting. If clustering is what's, I mean, how that's being driven is another question, but they nest in mass
Yeah.
in most areas. Maybe there's some exceptions, and the males sort of congregate and will mate with the females off the shores of the nest, I believe, and that raises a lot of questions about just kind of sperm viability, competition, that sort of thing.
Mhm.
And I wonder, I'm curious what you think about this, and maybe you could talk about some of your work with that. But if there would be some way you could use the morphometric measurements to get an idea for the viability of different male sperm over time. So if you took a measurement of, I mean, like fluctuating asymmetry, that
Mhm.
idea in biology, it seems like that's kind of been pushed to the wayside is not the best thing to analyze. But could you maybe get a measure for how successful certain males are with long term viability just based on measurements of how symmetric the hatchlings are?
Uh, that's a good question. Some people still use fluctuating asymmetry to look at issues of development, and for like hybrids that might have lower fitness. People look at that because it is still like sort of a proxy for whether things in development are going well or not. So I see no reason why you couldn't study something like that. It will be a little it wouldn't be easy because you would need to figure out exactly the to sample the male sperm, then do some crosses, figure out the hashlings, who has father which hashlings, which of the males have father which hashlings, then and unless you have virgin females, then it might complicate things. Although with genetics, you could probably tell apart if you know the fathers, like in a colony or something, and you know that the number of fathers can only be these 10 or 15 or whichever number of fathers there may be potentially. Then you could screen them genetically, look at the offspring from different females, look at their asymmetry, for example, and try and genotype those hashlings to figure out who is the father, and yeah, and check whether there's different fluctuating asymmetry for them. Never seen that done. So there you have a project that you might be able to do in the future.
Yeah, I just kind of when talking about the geometric morphometric stuff is interesting. It's like it's an interesting cross between nowadays with the neural networks, that artificial intelligence you can bring to a screen. There's so many different applications of that. Maybe something you could get at those population level genetic questions through analyzing characters like that. I mean, obviously the tough part of it is you'd have to track those hatchlings over probably a few
Yes.
decades to see who makes it, that kind of thing.
Yeah,
But
exactly. So you could that's the challenge with studying organisms that are so long lived, right? If you want to get at measurements of fitness, your typical study of 3 years, 5 years tops would only allow you to use proxies of fitness that are short term, early in life, or during a just very short period of time. and only a few people have been able to collect almost lifetime fitness measurements for populations that have been followed for 35 years or over. Some of the painted turtle populations, for example, but yeah, very few people do that because it is impractical for many researchers.
I do have one question. So when we talk about, I guess this would work for Podec nemis specifically.
Yeah.
When we talk about the competition, or I guess what my question is when you look at a mixture of potential fathers or something of sperm donors, is there a way to differentiate between stored previous sperm and actual direct competition within the same season?
Yeah, good question. Only if you have control setting, like say in a captive colony, where you have control over some of the matings, for example, so that you could actually run an experiment, a control experiment. Otherwise, for example, when I did the multiple paternity analysis of Podec nemis expans, I using genetics. With today, there's genetic techniques that are much more powerful than what I used at the time, but that's what was available at the time. Just typing a few genes, a few loci in the genome, and try to figure out the minimum number of males that could explain the genotypes of the hashlings. And with those data, there were basically some hashlings within a nest that many seemed to come from a father, and then fewer from another, fewer from another. And so that could be explained by multiple processes. One is that the one with the most hashlings happened to be a superior competitor. His sperm is superior to that of the other males. If all the males made it at the same time, at the same season, let's say, with the female, and that was just the best sperm. But it could also be that because turtles store sperm, that perhaps the one with the most hashlings, with the most offspring, was the one that made it at the latest moment from this year, for example, and that the one with fewer was a mating from last year, and the one with the few was a male that made it 3 years ago. And so maybe the sperm is used in the proportion that is present in the tract, which will decay with time. And so you will end up with these distributions that we couldn't tell if it was sperm competition or simply a matter of the timing of the mating, right? So you would need to do an control experiment, to be able to test whether it's sperm competition.
Yeah, that is a good point. I didn't really even consider about the viability of the sperm, especially over time. It would be hard to know whether it's just not super viable sperm that was recent, or if it's just something from previous encounters.
And there's not much done about sperm competition or viability studies in turtles, because it has that difficulty. If you have to do the best control experiments will be if you start with a female, when is a hashling, keep her separate, so that then when she matures and mates, you give her these choices and have different experiments with different number of males, different timing of mating, with different females to have replication. But we're talking about waiting for an animal that takes 5, 10, 15 years to mature until you're ready to run the experiment. So these are the challenges of studying long-lived taxa like turtles.
Right. And it something in one of the recent papers that has come out of your lab, that there was this may have been the painted turtle genome description prior to the actual analysis of the genome, but there was a sentence in there that stands out, with just that turtles are kind of an emerging model system. But it seems like so far in the conversation, you wouldn't think that in terms of ecology, things that have to play out on sort of a lifetime scale. But when it comes to the molecular side of things, and maybe we can kind of transition into sex determining systems for a bit, they kind of are, in a way, a model system to get at those complex evolutionary questions. And I, just sort of to start this conversation out, I think it makes sense to kind of step back and say, okay, turtles have this broad range of there's the, as you mentioned, genetic sex determination, temperature dependent sex determination. But within genetic sex determination, there are multiple different systems.
Mhm.
Some of them may be partially genetic, somewhat influenced by environment. There could be some mixing there.
Not in turtles, though. Turtles don't mix. It's one
Okay.
or the other.
Okay. So that's sort of a, overall. Okay.
Mhm.
But turtles have this wide array of different mechanisms for getting at sort of how sex is determined. And I guess just why? I mean, it seems like why keep all of that variation?
Very good question. And I don't know that we know that yet. When you do evolutionary analysis, phylogenetic analysis, and you reconstruct the ancestral trait for sex determination in turtles, it seems now fairly consistently, the more analysis that are done, that the ancestral turtle had temperature dependent sex determination, and that mechanism has been retained in the majority of turtles that we know of, and it has been lost 5 times at least that we know. So there has been at least 5 transitions from the temperature dependent sex determination, being the ancestral state, to a sex chromosome system, either XY sex chromosome system, and that's the most abundant, that has happened at least 4 times, and once to a sex chromosome system of ZZZW, sort of the type that you see in birds. So the XY and the ZW are different simply about in which of the sexes is the one that carries two distinct chromosomes. So if it's XY, the males are the ones that carry 2 chromosomes, just like humans. If it's ZW, it is the females that carry 2 different chromosomes, like in birds or butterflies. Why those transitions happen? We don't know. There's some correlation that we have observed of the branches of that tree of life of turtles, when in the lineages where there's a transition from temperature dependent to sex chromosomes, those transitions seem to associate with time of global temperature that were at its peak or was changing rapidly globally, when you look at paleo data of temperatures. So perhaps there was this ecological effect that was happening. Let's say if the world was warming for quite a period and warming fast, there was feminization of populations of turtles with TSD, the temperature dependent sex determination that we call TSD. And that feminization would have selected for any individuals who will produce the minority sex. And if any individuals had a mutation that was passed on to their offspring, so that no matter what the temperature, that female will produce sons, those sons will have higher fitness because they could mate with a lot of females, and so their genes of that mother will be represented at a much higher frequency in the next generation, and that will continue until the sex ratios reach a 50-50 level balance again. So perhaps something like that happened, and some of these lineages transition to sex chromosomes because of something like that. But we don't know. Is just the data are consistent with such a process. Maybe it was something else. Maybe something destabilized because something that is interesting that we also detected is that in the branches of that tree of life of turtle, when you see a lineage that goes from TSD to GSD, and there's 2 branches where there may have been a reversal back from GSD to TSD, a putative reversal. That when there's a change in transitions in sex determination, there's 20 times higher rate of evolution of chromosome number. Somehow there's an association between chromosome number change and transitions in sex determination. We don't know why that is. It could be just statistical artifact. It could be that there's actually a functional link between those two things, that when there are fusion or fissions of chromosomes, which will result in higher or lesser total number of chromosomes, that something about sex determination gets is altered, so that then the change in chromosome number causes the change in sex determination, or maybe it's the other way around. Let's say that the global temperature causes selection for this genotypic sex determination, and that happens through some mutations that alter the machinery of the the sex determination happens in more of a passive way, or that something in the environment causes the genome to become unstable, and that causes the fusion, the chromosomes to stick together or to break apart. And that mechanism that is causing that at the same time is changing the regulation of genes that act in sex determination. So it could be one is the first one, the other is the first. Maybe in some lineages it happened one way. In other lineages it happened through a different trajectory. We don't know. These are interesting observations and sort of correlations for which we have no definitive explanation.
Yeah, with these sort of evolutionary questions, it seems like you just kind of have to think about it from all different perspectives and then look for those link correlations and then
Yes,
kind of try to get a functional understanding of why that correlation exists.
exactly.
Um, when it comes to the genetic sex determination, I think that this is something where people will be very interested to hear. That is it TSD, I think, is more of a foreign concept as human mammals. That's very different than the human way of sort of doing of determining sex.
Yeah.
And so it's kind of a foreign thing, but genetic sex determination, I think that kind of gets placed in a basket for turtles, and it seems a little bit closer, but there's really a lot that goes into figuring out what the characterization of the sex chromosomes are, what types of chromosomes they are, actually using different techniques to find them. And you've
Mhm.
been involved in a lot of, with pluridyrs and cryptodyrs, actually looking for those chromosomes. And I'm curious, I mean, what kind of technology are you using to get at visualizing the chromosomes? What have been
Mhm.
some of the 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 things that have gotten in the way and the challenges in actually finding the chromosomes, the system itself?
That's right. So it is. So there's something that happens when sex chromosomes evolve. Like they will start like a normal pair of what are called autosomes. So autosomes are the chromosomes that are not sex chromosomes, and they always exist in pairs in our cells, because we receive half the number of chromosomes from dad and half from the mother, right? So you have a pair per a chromosome. They tend to look the same, any pair of chromosomes, but when one of those becomes a sex chromosome, usually happens because some mutation in it, in some gene, then is a sex determining mutation, that then any individual that carries that, if it is a male determinant mutation, that autosome becomes a proto Y chromosome, right, which is the one that is special in, say, humans to determine sex. The other one becomes, we will call the X. And over time, because you have this sex determining mutation, and now there may be, for example, other genes that are in that chromosome that might be better for males that could accumulate there, since only males, anybody carrying that chromosome with that mutation will be a male. So any other mutation that benefits males but might harm females, what are called sexually antagonistic mutations, will be advantageous to keep it linked with the sex determining gene. So when that happens and you have what is called reduced recombination, so these two chromosomes now don't exchange material during the production of gametes as any other pair of chromosomes does, Then mutations keep accumulating in that Y chromosome. Some genes lose function, some genes might be lost over time, and this original Y chromosome starts shrinking because it loses pieces. It first loses function, then it may lose actually genes or chunks of material. And that's how, let's say, mammals and humans, we have a Y chromosome that is much smaller than the X. So when you have that, when you have chromosomes that are of different size or shape, when you look at cytogenetically at the cells of males and females, in females, for example, all the chromosomes you find pairs that you see all the chromosomes during cell division. You stop them at what is called metaphase, when they're formed these nice little packets that you can then stain with just cellular stains and DNA stains. And then when you look at the chromosomes in the microscope slide, you can see all the packets. You can count per cell, then all the little packets. You can tell, oh, this is the largest, and this is the other one that is largest. They form a pair. These other two form a pair, and so on. But then when you do that in males, you start putting the chromosomes in pairs, and there's two that are left that are of different size and shape. Then you know, okay, I only see this in males, but I don't see this in females. Therefore, this is my XY pair of chromosomes. And then you detected the chromosomes like that, the XY. But sometimes in organisms like some turtles, the sex chromosomes are very similar in size, and hadn't been detected in the past, or are a pair of microsex chromosomes, because turtles have not only large ones but very tiny dot chromosomes. So how do you detect those? And for those we use modern techniques, because the older classical staining techniques couldn't tell them apart because they look pretty much the same, or people didn't look carefully. In turtles that we know have genotypic sex determination, and that you can tell not from cytogenetics. You can tell by doing incubation experiments and finding that every incubation you use in a wide range, you always have 50-50 male females. So when that happens, that tells you, okay, these ones, the temperature doesn't affect the sex. And if it's always 50-50, that means that something is segregating, is passed on in equal amounts to males and females to the offspring, that makes them either male or female in a similar amount. And that's when you start looking for this chromosome, this sex chromosome. So you do the classical staining. Do you see something that is of different size or shape? Great. You have your XY. If you only see that in males, or if you only see that in females, that will be called a ZW. But for the species of turtles for which you did the incubation, you know they're GSD because the sex ratios are always 1-to-1. But the classical cytogenetic techniques don't identify any difference in size or shape. Then what we do is the following. It's called comparative genome hybridization. You take the DNA of males and females. You label that DNA with different colors, red and green, for example. Then you take the metaphase chromosomes in a slide of bunch of males, bunch of females, and you paint them with these two probes that are red and green. Then you see where the DNA goes by seeing the red or the green that is painting all the chromosomes. If the content is the same, then you will have both red and green painting every chromosome. And what you're looking is for any chromosomes or region within some chromosomes that only have red or only have green. So if you painted the male DNA green and you see one chromosome pair that only stays green but not red, you know, oh, male limited, male DNA only content in that one. And again, you have an XY system that you identify. That's how we did the first identification of sex chromosome in Chelonia longicolis, an Australian turtle that has this micro sex chromosome system. The X and the Y are exactly the same in size, and they are these tiny chromosomes. You would never have found identified those as sex chromosomes if it wasn't for the use of these molecular techniques. And then we have used that was done in collaboration with colleagues in Australia, the laboratory of Jennifer Marshall Graves and Tariq Essas, who is now at a different university. And we work on another turtle and then another turtle, then some here that we did in my lab when we started doing the work here. And we have characterized several of them that are XY, most of them, and some ZW in soft-shell turtles, the spiny soft-shell turtles.
It's really interesting, just the difference in system. And some of them are sort of, I guess, backtracking that, like there's so much to unpack there. The reduced recombination that allows you to use that sort of the CGH procedure, I think that's sort of what's driving that, if I'm correct.
Yeah, that's just what happens evolutionarily, which is the cause of the differentiation of that Y or that W chromosome.
Right. And in terms of, I mean, I guess this is really what the question is, but that's just fascinating to me that the linkage sort of breaks between other, because that proto-sex chromosome is an autosome initially. Is that, and
Exactly.
so that linkage with other autosomes sort of disintegrates over time, and then that chromosome starts behaving differently. Is it known how that reduced recombination sort of spreads throughout the chromosome over time?
So one easy way that it could start is with an inversion in the chromosome. So in the chromosomes, you have the genes that are found in order along the arms of the chromosome. So when you have the autosomes in both members of the pair, they will be in exactly the same order. If an individual suffers a mutation where one chunk of several of these genes all of a sudden are inverted, and that happens during the formation of gametes, then that area doesn't recombine with the same genes in the normal autosome. And so having inversions is something that is a telltale, a little bit is an easy way to stop a recombination. And when you have that area inverted, then any other genes, these other genes that might be good for one sex but bad for the other, if they jump there or were already there, and then they are in this linkage that cannot be broken because they are, due to that inversion, they're not recombining anymore. Then the process that I described takes on.
Right.
And we have found those inversions in the turtle sex chromosomes. We found in Glyptemis, for example, the Glyptemis in Sculpta. We could see by the order of the genes that we were able to. We had DNA for little chunks of the chromosome that we painted with different colors, and we then painted in the chromosomes. And when you compare the order of those colors in the X of the Y, we could tell that there has been an inversion in the sex chromosomes. One first, an inversion that must have happened in the Glyptemis compared to the painted turtle that are close relatives. And then another inversion that happened in the Y with respect to the X. So, and those will be regions of low recombination, and is in those areas of the inversion that we found that green male-only DNA when we did the comparative genome hybridization. So it seemed that the process was ongoing the way that the models theorized that it should happen.
And so there's also been a bit of I think you looked at this with Chrysomes, the painted turtles. There were some hypotheses for a while that maybe even TSD turtles had some residual sex chromosome or chromosomal activity. I'm just curious what the search in that kind of led to, if that's the case, or.
So we looked at that because there have been some publications suggesting that the sex ratios that you obtain at the pivotal temperature and some other markers that you can measure at the pivotal temperature being in a TSD species, the temperature that produces even sex ratios, equal number of males and females. That was a sign that there may be a genetic sex determination that is hidden, overridden by temperature when the temperature is above or below that pivotal temperature. So we figure, okay, we have used the comparative genome hybridization to look for sex chromosomes. So why don't we use the same technique to try to see if there's any evidence of the existence of sex chromosomes in painted turtles? So let's just do exactly the same technique that we have done with genotypic sex determined turtles that we knew were GSD because of the incubation experiments. let's apply the same techniques to the painted turtle. And we had to first sit down and come up with a logic tree of all the possibilities of what type of signals are we expecting to see if they had GSD, if it was the male, if it was a female, different chromosomes, what will be the signals that we should see? And we ran the experiments, ran through the table of potential outcomes, and the results were we do not detect sex chromosomes in painted turtles. Now, that means at the resolution of these techniques, which is around these techniques of the comparative genome hybridization can tell differences that are at least 5,000 base pairs or larger. So the minimal size, the resolution will be, so we could tell that there was no evidence of sex chromosomes for anything at the resolution of 5,000 base pairs or above.
Yeah,
Right.
that's really interesting. I think just when we look at like future outlook, I think with turtle conservation specifically, aside from the destruction of habitat and things of that nature that we worry about, our shifting climate affecting sex ratios is something that is sometimes brought up. Do you ever find that, or I guess, is there any sort of control system to advert that risk within turtles, or is it something that is a major concern?
It is a concern. I think that the greatest threat remains to be habitat destruction and over exploitation, right, for turtles. But given that, if you have a population that is already small, then having a drastic a biased sex ratio can put them over the brink of extinction, right? And with most turtles, the warmer temperatures produce females. Having excess females is not too bad in a healthy large population. In fact, having an overabundance of females may enhance population growth, and that should be good up to a point. If you get to the point where there's so many females and so few males that finding a mate for females becomes difficult, then you're in trouble. Or that there's so few males that the females that exist are fathering all these offspring, that all of a sudden you're losing almost half of your genetic variation in a population because everybody's is becoming inbred, and inbreeding usually has a toll on fitness because through inbreeding, these recessive genes, so the alleles that of genes that cause disease for which you require to have two identical copies for the disease to express, you will find identical copies more easily if you mate with close relatives. So that by losing the males and having too few of them, then you will enter that danger zone of having the expression of deleterious mutations that can cause disease, like in the Florida panther that are so inbred, or cheetah that are so inbred that they have sperm deformities, deformities in the tail, their immune system will not function properly, and so on. So you will have developmental problems and other physiological issues. So it is a concern for that reason. And we are seeing with sea turtles, for example, that in some populations in Australia have become extremely female biased. So with TSD, you would expect that some populations might have more males or more females, depending on the region, the area, 1 year to the next. And a little bit because they're so long lived, they can withstand changes in the production of the sexes, too much for one sex, too much for another, 1 year after another for a while because of their longevity. But after, if it becomes too drastic, then that's problematic. And this population in Australia, for example, of sea turtles, are like 95 or 99 by now, all female. And they did genetic studies to figure out whether they were receiving individuals from elsewhere, and they're not. They're fairly isolated. So it is a population that is at a brink of going extinct if they lose the males.
I'm curious, just talking about what you spoke about earlier and how the transitions from TSD to GSD in turtles seems to have been correlated with peaks in climate. I'm wondering if there would be some way to forecast if you could have a reversal of that in the future if the climate keeps changing like
Mhm.
this. I mean, could you?
It is plausible, but it might be unlikely given the fast pace of climate change compared to the normal pace of evolution. Because evolution takes a long time, and for organisms that are long lived, it takes even longer chronological time. And the planet is warming much faster than that. So there's a constraint there. We do see, interestingly, that in the spiny softshell turtles that have a genotypic sex determining mechanism and have ZZ, sex chromosomes, when we look at the expression of genes during development, during the formation of the gonads, compare that to the expression of those same genes in painted turtles that have temperature dependent sex determination, we see a lot of thermal sensitivity still of expression of genes in the soft shell that are affected by temperature, even though the sex ratios are not. Some of that pattern of differential expression by temperature is identical to the one in the painted turtle for some genes. For some others, it has been lost completely. For others, it has flipped so that if in the painted turtle the gene had higher expression in males, now in the soft shell it has higher expression in females, for example. Even if it's not affecting the sex ratios, there's the potential that that type of sensitivity is a plasticity of the expression of the genes that remains in these genotypic sex determined turtles, could be the raw material that is selected in the future to reverse back to TSD. Because to reverse back to TSD, you need sensitivity of the genes to temperature. And if they already have that, then they could co-opt it again, use it if they lose the sex chromosome, if the conditions are right, like the selection is very strong, the conditions are perfect so that they will transition back to a TSD mechanism that they could do that. So we see the raw material for it there, but it's hard to predict whether they could use it at all or not, again, because of the pace of evolution. And a bunch of conditions would be necessary. But certainly having an almost entire female sex ratio, female population, that's the type of condition that will have a very strong selective effect for any individual who can produce males no matter what the temperature in a population like that.
Interesting. Yeah, it's just okay. So thinking about that and sort of the genetic network that underlies TSD, I think that's kind of a good transition into that. Is just it's fascinating. A thermal stimulus can kind of be transmuted into some physiological change,
Mhm.
and there's this in mammals. There's a diagram that reappears in a lot of your research that seems to in each consecutive publication it gets a little bit altered, a little bit more detail, that sort of thing. And the mammalian circuitry that underlies sex determination with the reptilian homologs and the relationships. And I'm just curious, I mean, what are the, because early on there's kind of a subset of genes that SF1, DAX1, these different genes that are involved in this. And I'm curious how you knew to look at those. And now, I mean, what with transcriptomics, these things? What have
Yeah.
you learned?
Yeah. So when I started looking at that, it was okay. Let's start doing the developmental biology of sexual development and gonadal development in turtles. Where do I start, right? With which genes? And at a time when what was easier to do was to interrogate one gene at a time, then I had to find which genes to choose. And the way that uh, I did and people normally would do is, what do we know from other organisms? So what do we know from mammals or from birds? And these genes, SF1, WT1, DAX1, aromatase, DMRT1, were coming up over. They were the genes that were studied the most at that time. So it was a logical step to start there. Let's start with candidate genes that come from other vertebrates that we know are important for gonadal formation. The first thing, do they exist in turtles? And yes. How do they get expressed by temperature? Are they affected by temperature? And we found, looking at different developmental stages of gonadal formation in painted turtles and in the soft shells, that they had a differential expression. Then the next question that one would ask is, how are they sensitive? Are they sensing directly, or is there a gene upstream of those that is causing what we see as a sensitivity to temperature? So then you start going upstream and upstream to try to find the earliest time point and the earliest element that might be the one sensing the temperature and translated that signal into opening up one developmental pathway or the other for gonadal formation. And so that we could do later when transcriptomics became viable with the budgets that we had and with the study system that we had. So the transition to transcriptomic was so that we could go from the genes of the mammalian network that was very well characterized to interrogating the turtle in sort of an agnostic manner, just letting the turtle tell its own story, and not forcing the story through the lens of the mammalian models. And at that point, we saw that those elements were still important. They still had differential expression, but there were a variety of other genes that were also thermosensitive.
Right. Something that stood out to me just in terms of the layout of the network in that the diagram is that it seems like the male, there were the AMH, DMRT1, GHT are all sort of directed in upregulated in testis development. Whereas in the female, it seems like more of a simple pathway that the testosterone has to hit a certain point, and then that aromatase gets upregulated, and then that leads to a cascade that produces ovaries.
Mm-hmm.
I'm curious that it's not an imbalance, but why is it that the male system requires more pieces to it and the female system doesn't?
I'm not sure that it does necessarily. It is that when we interrogated these genes from the mammalian point of view, that's how the data came out, right? But now that we have the transcriptomes, the networks are fairly similar in terms of components and the complexity of them. It is true that some genes that are important for the formation of the early bipotential gonad, like SF1 and WT1, for example, they are important transcription factors. So their job is to activate other genes. They do that early on to just make the little gonad that then is going to make the decision to go the ovarian or the testicular pathway. And they also are important later on they're masculinizing. So later on they will be important for testicular formation and for maintenance of that of the organs. But the two gonads, they need elements to be maintained as ovary as testes throughout life. Aromatase is a hormone that is very important and critical for ovarian formation. That was known from early on. Just work on the protein itself, then on the expression of the gene that codes for it, and transcriptomic studies, that definitely is crucial for ovarian formation. So that keeps coming as an important element. And you also have then the male hormones that are important for testicular formation with those other. But yeah, I wouldn't say that one or the other pathways is more complex. And at the beginning, in the bipotential gonad, there's the two male and female pathway elements. There's some elements that are equally important and is sort of active. It's not that one is active and the other is not. Both are active in a balance that it stays right there, and then something about the temperature then tips the balance one way or the other. But there's elements that are active at the beginning, like just ready to go. Tell me, left or right. And then once they receive that information, they go one side or the other.
That's interesting. Yeah, a lot of it, that my understanding of it is based on that older diagram that was, I mean, it kind of highlighted the reptilian homologs within the mammalian system. But yeah, I guess with the transcriptomics now, it's just, that's become so, you can look at so much at once. It's just these heat maps of what's upregulated. So, I mean, has there been a revised network diagram incorporating all those components that's been put together?
We're working on one of my PhD students is working on a network for the painted turtle and the soft shell turtle, as we speak, is part of her dissertation. Wow. So that we can do that by contrasting two species with and without the temperature dependence, and doing transcriptomics and looking at protein DNA interaction, transcription factors, and so on. So we should have something revised fairly soon. Yeah.
That's exciting. It's just kind of fun to visualize what's going on. You know, you can see an organism and kind of see what went into that to some extent, and really fascinating. Well, and so there's, I think that within the realm of the TSD, there's also the thermosensitive period, and we've touched upon that. But why is it that there's only a period where the temperature matters, and it's not the entirety of incubation?
That's an excellent question, and I'm glad that you ask it that way. We don't know why, but it happened. Perhaps it is that way because it takes a while to form the gonad, and once that bipotential gonad exists, then is sensitive to temperature, and it can go one way or the other, and then differentiation. So you have, there's different steps, right? You start with a single cell, you start growing an embryo, then what is going to be the renal portions, the adrenal and the gonad form. The embryo has to first specify the field where that's going to happen, to say, okay, this is an area where we're going to build those organs. Then you have the gonad, a ridge that forms, a gonadal ridge that forms, and that then matures a little bit to be this bipotential gonad, and all that takes time, right? We're talking about organisms that the turtle incubation for, say, painted turtles can be 45 days or 90 days, depending on the temperature, right? And so that takes time. Then once you have the bipotential gonad, then you have the period where they are more sensitive to the temperature effect. The first thing that needs to happen, if it was a mammal, the same is happening, but it's happening perhaps in a shorter period and it's not dependent on temperature. But the first thing that will happen is you specify the field, then you get the bipotential gonad, then that gonad commits to becoming an ovary or a testis. That's the determination. Then it has to differentiate into an ovary or a testis, and that takes some time. In mammals or other organisms with genotypic sex determination, that may be linear. Specify, determine, then differentiate, and then grow. in turtles or with TSD, for example, then you have the bipotential gonad is formed, then the determination, that thermosensitive period takes weeks or more, because it happens to be sort of in the middle third of incubation. Why exactly? Maybe it has to do what I just mentioned, that it takes time to get to the point where they can actually do something with the information from the temperature. Then the determination takes time, because during that thermosensitive period, if you were incubated eggs at a all male producing or all female producing temperature, you could shift them and still and change the sex ratio, which means that that there's flexibility, there's plasticity within that time, which means that the determination, the commitment is not final, and it keeps like, I don't know, I'm not making the decision yet as a my potential gonad. At the same time that some differentiation of the gonads start happening. If there was more accumulated signal from a male producing temperature, then starting to go the testicular way. But if you were to shift them to a female producing, then that the differentiation that was happening will reverse and you end up with an ovary. So there's more overlap between the determination and the differentiation, which is also an interesting difference. And then it gets to the point that enough differentiation has occurred and the determination ends, so that what is left is continue differentiating to make a solid ovary, a solid testis that will be at that point irreversible unless something drastic happens. But in terms of temperature, it will be irreversible. It is interesting, though, that in some taxa, I observed this with Porognemis expansa. Some people observe something similar with some Australian turtles, that the temperatures experienced before that canonical sort of thermosensitive period explain a little bit of the variation in sex ratios that we obtain from experiments. And that suggests that perhaps something is happening early on that matters for sex ratio also, but that we haven't paid attention to this non-canonical period before the more known, better known thermosensitive period happens. And it could be that it just puts the individuals in a growth trajectory that somehow makes a difference, or something. We don't know why, but we have made those observations. So there's something there as well.
Right. It's almost like sort of an enzyme cascade. It's you need to get to a certain point and then the system will fall with so much force behind it that you're not going to sort of change the trajectory of the sex.
Yeah, we do know from just temperature and sex ratio data. We do know when you look at incubation temperatures and developmental rates, we do know that the effect of temperature is cumulative. And so that there's sort of similar to what in plants are degree days, something like that happens with these animals. And in the end, when you just look at thermal profiles and sex ratios, particularly in natural nests where the incubation temperature goes up and down every day, you notice that what matters is how much development took place at a low temperature that, if it was constant, will produce only males, or at a temperature above that pivotal that, if constant, will produce females. So depending on that, that's how you end up with that cumulative effect and developing a testis or an ovary for these turtles.
This is something, again, as you mentioned earlier, Dr. Georges was one of our previous guests, and that was at the time I was doing some things in class with trigonometric integration and trying to kind of figure out an application for that. And it worked perfectly because that was his looking at the thermal variance over time was like, this is perfect. A lot of calculus stuff people
Absolutely.
It's tough to find why does this matter, and something like
Hmm.
that applied to biology was cool. But regardless, that brings up another question, and then maybe we can kind of transition into a few other things. And but with climate change, I mean, I was recently working at a field station in Belize, and they modeled some of the climate data. And over the past 25 years, you can see that the temperatures on a daily basis are start fluctuating much more,
Hmm.
and this thermal variation. I mean, is that so? That again, going back to what you were saying, it's that amount of development that's occurring at a higher temperature, even if it isn't the mean temperature changing, it's just that fluctuation could be
Yes.
enough to push
Yes.
feminizing temperature.
And we did an experiment with painted turtles that we published some years ago, I think in 2019, where we noticed we were experimenting precisely with the temperature, keeping the mean and then playing with the temperature. And we did, and unfortunately, the results are a little bit troubling for conservation of turtles with TSD, because what we found was we started with temperature that was constant male producing or constant female producing. Then we did very simple artificial incubation experiments, um, add plus or minus 2 degrees or 4 degrees or 6 degrees around, but keep them in the same. And what we found was that with that first set of experiments, the male producing mean end up with the plus or minus 6 degrees. So when the fluctuation was very large, feminized the eggs, but with the female producing temperature, the greatest fluctuation masculinized the genes. And so, oh great, this is going to solve that. This is how the animals are going to survive, because the temperature will go up, but the fluctuation would increase. If that is masculinizing, they will produce males. Great. Now that was very simplistic, 12 hours high, 12 hours low. How about natural nets? So we recorded the profiles of temperature in the field, look for some that produce only males and only females. Then we took a male and only male producing profile from the wild that was going up and down, shifted it up as if only the mean temperature increased with global warming, and that increased the number of females. Then we started stretching the profile to simulate the type of other experiments we had done with the 12 hours up and down. And when we did that, um, starting with the male producing profile, you shifted it up, you produce females. You increase the fluctuation, you produce females instead of males. When we take a profile that produces only females and you stretch it, then you either keep getting females or the embryos die, because now the excursion at very high temperatures will kill them. And if it didn't kill them, then the other ones just remain to be female producing. So all of a sudden, we realized that there was no good news, that what might happen is that with global warming, the mean goes up, is feminizing, the fluctuation of temperature goes up, is feminizing. So there will be an acceleration of feminization or demise of some populations if they reach into the mortality zone of very high values. So yeah, that was we thought we had good news, and in the end, it didn't seem that that was the case.
Right. And with the realm of just, I guess, temperature dependent sex determination, that those gene networks, there's the epigenetics. The field of
Mhm.
epigenetics is something upcoming, and I recently took a genetics course, and they went into that a lot more than I would have expected. Learning about a lot of this stuff early on, and like middle school, high school. That it wasn't really even in the conversation at that point 10 years ago or so. So
That's right.
it was cool to see that. But regardless, this is something that your lab has looked into in terms of
Mhm.
how does this influence the gene regulation for TSD and other things. And I'm curious, I mean, methylation, those sort of things, how is that factoring into sex determination?
So we have looked into DNA methylation in particular, and some of the we found that the hashlings of painted turtle male and females have different methylation. And so, and when we did, just like transcriptome looks at all expression of genes, the methylome looks at all methylation of all genes in the genome. We found several genes that were differentially methylated between males and females. And some of the genes that were differentially methylated, if we looked at their expression at the later stage of development for which we had the transcriptome, stage 22, some of those genes in the developing testis or developing ovary that were, say, more highly expressed in males were hypermethylated in females, and the other way around. Some genes in the other sex and were, so what that told us is that some of the differential expression of these genes during development, it may happen not because one gene is activated to a higher volume, but because that gene is silenced in the opposite sex through DNA methylation. So this is a way of regulating gene expression. So the question is, how early does that happen? How is that occurring earlier in the sex determining period and the thermosensitive period? But I think about epigenetics, we have now a very just beautiful data coming from Blanche Kappel's lab that is that has looked at the more upstream some elements and the potential epigenetic regulation of TSD with slider turtles, with trachemies. And the system, the hypotheses that come from that, the model seem to be fairly good and explain a good bit of how TSD actually might work. And it's also epigenetic, but it's not DNA methylation. It's through methylation of histones. Histones are the proteins that the DNA wraps around when it's packed in the nucleus in the cell. And depending on how compact they are, then DNA will be inaccessible for transcription, so the genes will be silenced. But when those it unwraps a little bit, then it opens up and the genes can be read and produce the proteins that they code for. So what they found is that in the Trachemys turtle, at the cold temperature, there is that cold temperature activates the expression of this one gene, KDM6, that produces a protein whose function is to take one of these histones that the DNA wraps around and take away 3 methylation marks that histone has, because when that histone has the 3 methylation marks, then it makes a difference. It's sitting next to the DMRT1 gene, which is one of the important genes in the TSD cascade. And so when that histone is 3 methylated, it represses DMRT1. It can repress DMRT1. When DMRT1 is repressed, then the female genes are active. When that at cold temperature, those 3 methylated marks are taken away, then the DMRT1 gene is active and it can activate the rest of the male pathway. At warm temperatures, what happens is that there is phosphorylation of a different proteins, that 3. When that's phosphorylated, it represses the KDM6 gene, which is the one that did this other business. And by repressing it, it represses the taking away of the methylation marks. Then that stays in the promoter. That histone with the 3 methylation marks stays in the promoter of the DMRT1 gene and stops the male pathway and activates then the female pathway. So that's the what we have now as the best model as to how TSD works and how the signal of the temperature may be translated into these testicular or gonadal developmental pathways.
That's really interesting. Based on at least the old sort of diagram with the mammalian system, it did seem like there was kind of a lack of repressor signals on a lot of things. And I was a bit confused with how, I mean, what is the thing that's controlling the production of the hormone or the gene that's
Mhm.
causing expression of this that leads to the
Yeah,
ovarian or the testicular development,
yeah.
right? If you add something that's sort of extra genetic, it's external and kind of doing architect in well, methylating that sort of thing and
Mhm.
controlling the expression level. That
Yeah,
makes a lot of sense that can just
exactly.
sort of, yeah, cause those cascades.
Yes, exactly.
Well, I think maybe we can sort of the one element of your work that's really fascinating is that there are some, again, it takes into account a lot of different areas of science, but there are also a lot of takeaways and things that can be learned and applied to different fields.
Mhm.
And biomedicine, therapeutics, these sort of things are really germane and can learn a lot
Mhm.
from what you're doing with turtle work at the genomic scale. And one of these things, the painted turtle genome, was a big project that you worked on, and I think 60 or so other people
Yeah.
at least. And um, I'm just curious. I mean, what was it like to undertake that project, and what were the things that you learned? And do you think that there's potential there to harness some of the specifics that you learned from the painted turtle genome in human therapeutics?
Mhm. So that was a project spearheaded by Brad Schaefer, and yeah, there was a bunch of people involved in that because it was a big task, a big undertake by then. Right? Is It took a while because the technology required mixing different technologies. It was a slower pace than today. Today you can get a genome sequence like that.
Yeah.
Is completely, it has changed so much in these 11 years or so since that publication. And so we learn uh, important things about the painted turtle and about how to deal with the assemblies of the genomes. That was a very important thing that we did. So we later improved that genome in my lab using some of the cytogenetic techniques also, to be able to locate, to be able to tell the sequences that had been obtained from the assembly of the genome, where they live in the chromosomes. And so we did that physical mapping that is called, and later we did some other types of scaffolding with other techniques to try to improve the contiguity of that painted turtle genome. We have used that genome for other projects, like the development of the organoids that we did with painted turtles and other turtles. When we characterize, and now when we do any genomics, mapping any sequencing data, transcriptomic, that we have two good genome assemblies is essential. So we learned from that painted turtle project so that we could do better genomes, and we're working now on several of those, two of which we hope to publish this year, and some other coming in the next year. And how to assemble them in a better way was one important thing. But then, how do you use them, right? To identify regions of interest, gene of interest, and for the case of biomedicine, painted turtles are very anoxia tolerant and freeze tolerant, and they super cool. And so when you look, there's a lot that we can learn by looking at the physiology, the molecular markers that are overexpressed or repressed in during anoxia or freeze tolerance, and so that we can figure out what genes are involved in that, right? And some of those genes were identified in that first painted turtle genome paper, because there were some of our the co-authors did transcriptomes of under anoxia to figure out what genes were overexpressed. So those are genes that can be studied um, more now. But once you have candidate genes for anoxia, for example, or for sex determination from the work that we were talking about, at some point, what you want to do is to be able to validate that the genes actually are as important as you think they are based on their expression levels. And how you do that, you need to manipulate the gene and see if you get the expected response if they were really important. For turtles, doing that is very difficult to do because they are long-lived again and they are seasonal breeders. So doing experiments or transgenics the way that you would do for fruit flies or worms, the elegant worms, is not feasible for turtles. So one way around that we worked on was to develop organoids so that you have a mimic of an organ, simplified mimic of an organ that you can grow in a petri dish, and then you do the transient transgenics there. Then in that mini organ, then you can manipulate the genes and figure out if you get the response that you are predicting. So for anoxia and the cold tolerance, the liver organoids that we develop from painted turtles, from soft shells, and from snapping turtles, those become important tools for the scientific community to use to get at those tests. So in that paper of the painted turtle genome, there were a series of genes that were identified. One of them was notable. It had very, very high overexpression under anoxia. So now we have the organoids, for example, to look at this in the liver, to take that gene. And we also developed the tools to do gene editing, so to knock out a gene, to delete it from the genome, or to silence a gene or knock it down, the expression, and or activate one by overexpressing it. So we developed these tools that now we're in the position to start taking, say, that gene that was a candidate from that 11 years ago, manipulate it, make the anoxia experiments, and see if you get the response that you predict, if it's actually a gene that is important for anoxia tolerance.
That's, yeah, really interesting. Something that stood out to me in that paper was there was a mention of a gene, the SLC2A1
Mhm.
gene, that upregulated the GLUT1 transporters for glucose transport under anoxia, which it makes sense. I mean, you're going to have to do that to power the cells and under that kind of stress. But there was a sentence in there that said that this could be something useful for GLUT1 disorder in humans. And
Exactly. So for example, if we are able to understand how a gene like that functions, for instance, how it regulates, so that type of those genes that work for in the glucose pathway, when these turtles undergo anoxia or the supercooling or the freeze tolerance, what they do is they undergo hypometabolism. They enter hypometabolism, so they reduce their metabolism to a minimum, and to do that they need to regulate the glucose pathways, which are the pathways for metabolic energy production, right? And so if you learn how those genes work and how you can manipulate them, then perhaps that leads to something for therapies for diabetes. But the anoxia itself, for example, or the cold tolerance, could lead to, if we understand how the turtles survive those harsh conditions, which are conditions that a human would experience after or during a heart attack, or liver cirrhosis, liver disease, or then or lack of oxygen to the brain or so. So if we understand how the turtles are able to survive living without oxygen for months at a time or at very, very cold sub-zero temperatures, then learning how they do it, we could inform to figure out, is there a way to develop a therapy so that we improve the survival of people after a heart attack or after liver cirrhosis, or during a surgery when you need to transplant organs that requires to cool down the organs or keep the organs without oxygen for a while, and then when you put it back in the patient and the recipient, then you start a flow of blood that is going to cause a rush of oxygen. And usually the injury occurs not just because you don't have the oxygen, but when there's the reperfusion, when the oxygen comes back and there's damage from too much oxygen. So understanding how an animal that is resilient to it manages to do that, then you could start helping develop therapies for humans, because the regulation might translate in some ways.
It's really interesting how sort of you started out studying ecology of the Podocnemus, and it sort of evolved into something that has all of
Completely.
these interdisciplinary takeaways
Mhm.
that could be saving lives. And,
Mhm.
it's really a cool thing. Um, so it like that is something that using, so you seem to sort of believe in this, is the power of using animals, but power is maybe not the right way to put it, but the realized ability to use the molecular architecture of animals to inform human medicine.
Mhm.
I mean, that's it's a really interesting field, and cool that turtles could be sort of directly um, saving lives at a broader scale.
Yes, because in medicine, you're trying to find solutions to things that nature already found a solution for. So it's sort of using the power of evolution. Evolution already ran the experiment. Let's look at nature, see what that solution is, and can we biomimic, right? That solution into humans.
Yeah, that's a great way to put it. That's really interesting. I haven't thought about, yeah, from that perspective, it makes sense. Yeah, use the information, and it's probably applicable in some way, even if it's
Hmm.
different. We
Yeah.
just have to find it. Is there, for cancer research, is that something? I mean, turtles, the Peto's paradox idea that for how big they are, they rarely get cancer.
Hmm.
Is this something that organoids could be useful for?
Absolutely, absolutely. Developing organoids from papillomas or the cancers that the few cancers that turtles have, or looking at the genes that are oncogenes, that are cancer genes in humans, and see how they're regulated in the turtle. Yeah, that those absolutely could be areas of research that are, yeah, promising avenues, looking at how they regulate cell growth or division, and absolutely, yeah.
It's awesome. Well, maybe we can sort of, there's some level of things that are fun to talk about, but don't necessarily make it into the academic papers and that sort of thing. But I'm curious, out of all your adventures, what's been your most memorable adventure in pursuit of
Mhm.
turtles or research related?
Yeah, there's probably multiple ones, but during my PhD, I had very limited funding to do all the field work with the Podognemis turtles, and I had to run all the field experiments in one season and one season alone, because that's all the money I had. And the year I was going to go 1 year, and that year I didn't get the funding in time, and thank goodness I didn't, because that year there was flooding of all the nesting beaches, and every single nest that season died. So I went the following year, but concerned that perhaps the same thing could happen. We had a field station that was a little uphill from the nesting beaches, right in the riverbank. And so I decided that we needed to build this artificial nesting beach in the right by the where we were living, where we were sleeping. And that meant the experiments that I had set up in the natural area, in the nesting area, were 2 squares of 3 meters by 3 meters. So like 9 feet, a little like 10 feet by 10 feet, and over 3 feet deep. So we had to dig up that much soil at the campsite, then go to the nesting beaches, get that much sand to then take it in a 50-pound or 80-pound sacks in the little boat from the nesting area to the campsite, up the hill, dump it into these humongous holes that we had, and we developed this bamboo, like PVC pipes, but with bamboo, so that we could drain any rain that will drain from the bottom of this hole so that we wouldn't get inundated if there was too much rain. And the amount of work that that entailed was just absolutely humongous. I was never in better shape and strength than during that time. And fortunately, I had help of a good friend and colleague and another good collaborator from the area. And we were able to do that. We didn't need to use them, but the work was absolutely, yes, an incredible amount.
That's nice that it didn't end up being the funding was late. I'm sure it was frustrating initially, but
Yeah,
hindsight,
the blessing in disguise.
yeah. Well, also, I mean, it's been interesting to speak with. We've had conversations with a number of folks like yourself doing work with genomic data, these sorts of large scale operations that require a lot of different areas of expertise to come together. And I'm always curious what the most challenging project for you to work on is, and if that has been the most rewarding thing, if there's a correlation between those two things.
Well, the getting into the molecular side of things, that was definitely a challenge because that was not my original training. And so that meant that I had to retool myself to go into developmental biology or going to genomics. So for my PhD, I did the ecology, thermal ecology part. I did some basic population genetics and ecological genetics, and that enticed me because I always was interested in evolution and genetics. That when it got to the point that I couldn't quite explain how the males and females of Porognimis expansa were produced in the wild by the thermal profiles, I thought I need to just look under the hood and get to the molecular basis of it and get to the developmental biology. And that was there was a lot of fear of, can I do it, right? And this is not my training, so that imposter syndrome of, I'm diving into a field that is absolutely new to me. But I took the challenge on and did it. So it was very rewarding in the sense that what what we're doing now, all the molecular and genomic stuff that we have done, the development of these organoids now, this is like science fiction for when I started. And sort of being able to get to that point, not losing track of the bigger picture of the ecology of the animals, of the what happens in the wild, because I think one needs to keep those things interconnected, right? Because it's ultimately they live out there, experiencing conditions. The natural history is important. The ecology is absolutely essential to understand the rest of it, to go dive into the cell molecular level. But yeah, it has been very rewarding to do, to be able to get to that.
Yeah, it's a really interesting trajectory. As I've already met. Yeah, starting with the whole animal,
Mhm.
getting interested, and then diving into the internal part of the animal, and now developing things from that
Yeah.
internal understanding is really
Mhm.
a cool thing. And it's true. I think what you're saying makes a lot of sense. Like there's two different routes. The ecology route again is that's the animal, the environment, and then there's the more analytical route. And I'm starting to see this coming up in the two areas. And there's a lot of we can model this, whatever, but you can't forget about
Yeah.
the actual nature because a lot of times that will be the exception to your model and you
Mhm.
have to keep both things in mind. That's yeah, really interesting. And so I guess just to finish things off here, um, just for us as undergraduates coming up and really interested and motivated to do turtle work, and for a lot of our listeners that are younger, in college, different levels of education, what would be one piece or a few pieces of advice you'd give for someone looking to make turtle work a career?
Hmm. Good question. And I think the most important part is at what level do you want to study the turtles, right? What's your interest? Find your passion that way. For some people, it is natural history, and that's extremely valuable to do. We need more and more of that. For some people, maybe behavior, maybe the ecology. For some, maybe the evolution. For some people, maybe working with museum collections. For some people, maybe the molecular part, or phylogenetics, or so there's so many aspects that you could study turtles, that I think that will be the first question to ask yourself. And do you want to do that work, or you're interested in conservation, for example, essential for a group of organisms that are in peril like turtles are? And do you want to work local, or farther away? And there are a lot of different types of jobs that people interested in turtles do, right? There's the people working in, say, the DNR agencies, or in conservation NGOs that do rescue of or protection of nesting areas, be that sea turtles, for example, that's common, or people who do population monitoring, and so on. But I would say that something that would be nice to do, and I don't know that we do it to the extent that we could, is to, for people working at different levels, to interact more, so that we can leverage the expertise of different individuals, so that um, perhaps you don't need to learn everything from conservation to molecular biology, right? But you could speak to each other, and then leverage the expertise of each other, so that you get more out of the work of each person, so that there's an emergent property and emergent knowledge that comes from that. So creating coalitions, keep the communication up. And nowadays, with things like this, right? There's the virtual world that we can remotely interact with each other without having to travel to India, or to Africa, or to South America to talk to colleagues. We could do that remotely, and perhaps coordinate a little bit more some of the research that is done, so that we can do comparisons that apply to a broader range of turtles, if is done in a more systematic way. Um, those will be, yeah, things that I could think of that will be helpful.
That's yeah, right. Just the creating, especially in a digital age. I mean, that really hits home for me. Is there's no reason not to be connected with
Mhm.
as many people. So we'll take that advice to heart, and for people listening, for sure. Well, I mean, this has been awesome. If there's anything you'd like to finish off with, or anything, I'll open the floor to you.
Yeah, well, just thanking you for the invitation, and this is great. Your enthusiasm and your love for turtles, and creating this space for people to be able to learn and disseminate what we've learned, and that's just a fantastic platform. So I'm very happy that I was able to participate.
Yeah, thank you for giving your time, and it's been awesome. We like to finish off with a quick round of turtle trivia. I don't know if you have a few obscure turtle facts you want to
Oh,
throw at us.
eh, I didn't think about this, the trivia part. Obscure factoids.
It's always tough on the spot, but
Yes, yes,
yeah, I
yes.
mean, we could throw one your way, or if you want to come up with one and toss it to us. However,
So I ask a question and you guys answer. Is that the idea?
Mhm.
yeah, however you want to do it. Yeah, that works.
So what's the longest um, incubation? The longest that incubation time for a turtle?
Oh, that's a good question. I think I remember this within a plus or minus 10-day range. Uh, I'll throw out at 648 days for Cheloniac expansa.
Hmm.
Is
Yeah, well, so yeah, I don't know the exact figure, but it's months, right? That these eggs wait
Yeah,
to
more.
hatch.
Yeah, almost 2 years, right? I think it. Yeah,
Yeah,
pretty crazy. And all the diapause, the aestivation, the whole thing.
exactly, exactly. Yeah, that when I learned that factoid, I thought it was really unbelievable.
Yeah, that's really interesting. There's a photo in John Cann's Turtles of Australia book of a one of the large snake necks dug a nest in a floating styrofoam plant box
Oh,
sort of thing that was in a lake. I found that pretty interesting that it nested in that, it's
yeah.
but
So
uh
an observation, a curious observation that we made in the Porognimis expansa, in one of the nesting beaches that I showed the sandbars where they nest, there was in one season a flooding, and there had been a lot of nests that had been flagged. Then the flooding came, and when the water receded a couple days later, all the nests were gone. They had been dug up by fish.
huh.
And i and that struck me as, I just hadn't thought about that. I hadn't thought about how important of an energy source these animals were in their ecosystem.
Right.
And it made me think that that type of flooding happened often during all the millions of years that these animals have lived. And that in the past, Porognims expansa in particular, when explorers like Humboldt and so went through the Amazon basin, and they described in some areas calculated like 100,000 nests or more, and describe this migration of animals, that there were so many that you could potentially walk on their shells between the side of the river and the nesting areas and the middle of it, just cross a river stepping on one and another animal because there were so many covering the water. And I thought, if that was the case and flooding happened in some years, the amount of energy of just cycling energy through the ecosystem that they provided was unbelievable. So they are in danger, and the populations have crashed in some places or very small in others. What was the cascading effect of their loss to the rest of the ecosystem, right? To the trophic chain. Did fish suffer because they didn't have that resource? And they also eat the hatchlings when they come to the when they get to the water. There's birds that pick them from in the beach. There's some insects that eat them inside the nest. Then they come out and the birds are waiting for them. Then they get to the water and the fish are there waiting to eat them. And from there until they really grow and mortality goes down. But losing going from nesting beaches that had, say, um, 100,000 individuals to only 5,000 nowadays, that has had to have a toll in the rest of the ecosystem that I don't think anybody has measured.
Yeah, right. A question of how robust and flexible is the whole ecosystem,
Hmm.
and how many sort of saving loops are built into it. But
Yeah,
that's really interesting. That reduction of 95% or so in a lot of those places. What does that
exactly.
do?
Yeah,
Right.
and who else? Perhaps some other populations crashed too that nobody was paying attention, of some other organism out there that depended a little bit or a lot on the energy from the turtles.
Yeah, and what are the secondary impacts on human populations in those areas? Are those areas, are there a lot of indigenous people
Yes.
around there? Okay, so interesting.
in some areas in of the Amazon basin. In some others there are not, but yeah, in some areas there
But
are.
around where you studied, there
There,
were.
yeah, and in fact, I work within an area from an indigenous community that were very gracious and to allow me to do the work there. They were interested in conservation, and so was I. So it was a good partnership in that sense. Their consumption was, if it was just for their own consumption, then that wouldn't be too high. It is the commercialization that for markets in Brazil, for example, where most of the animals get sold to, that's what causes more of a crash. And traditionally, they didn't consume the Porognimis turtles, but later the taboo around consuming them sort of disappear, and then they started consuming an animal that normally didn't have an impact. Human related mortality was not, anthropogenic harvesting was not a thing until settlers came and started that practice.
Right. That's I in Ed and Don Mole's River Turtles book. There's the St. Creek reported a lot about that, and yeah, that's just. I mean, with all the other bad things that the early European explorers did in the area, that was one that, yeah, creating a effectively commercial scale
Hmm.
market for the Podocnemus. Just, yeah,
Yeah, it's unfortunate.
yeah, not good. But yeah, that's cool that you've had experience working there and have seen those fascinating animals in the wild. I mean, that Arabata must just be incredible to watch,
Yes,
even if
yeah,
it's
yeah.
produced.
They're beautiful. They're absolutely majestic. Yeah,
Yeah, well, that's awesome. That's probably a good place to sign off. And
yeah. Thank you so
uh,
much.
but yeah, thank you for everyone
Yeah,
out
thanks
there.
for coming on.
Yep,
Absolutely, my pleasure.
we'll see you on the next one.